Yep, you're good to go.
Oh, brilliant. Okay. So I know you probably know about SAMS, but just to remind you, we are Scottish Association for Marine Science. And as the name suggests, we work on anything related to marine science, from bacteria all the way to robotics and drones and anything in between. We just, I learned 20 minutes ago, published our research strategy that you can visit on our website. We are also associated with the University of Highlands and Islands, so we offer a variety of degrees, including PhDs. We have a commercial branch as well called SAMS Research Services, or SRSL, that does a lot of consulting, surveys, modeling, and all that jazz. And we're also home to CCAP, or a culture collection of algae and protozoa. So what is CCAP? Some people call it botanical garden. I call it the seed bank of algae. Nevertheless, we have more than 2,500 cultures that you can purchase, which is quite amazing. So these cultures are microalgae, macroalgae, cyanos. We have some pathogens as well. CCAP offers a variety of services as well. We have trainings and workshops and analytical services. We do cryopreservation. We also have a new service for patent depositing that the ladies wrote a paper about. There are two big projects that you may be familiar with. One is the Genealc, which is looking at boosting seaweed economy via genomics. And the second one is the Tree of Life project, which is sequencing genomes of microalgae in Britain and Ireland. Not all of them, just some of them. So CCAP is made of algae and people. So as you can see, we have a whole army of people that are ready to help. I think among all of us, we have more than 100 years of experience. So we are really ready to help. And if we cannot help you, we probably know someone who can. You probably noticed that one of the pictures is not a person, it's a creation by Rachel. It just makes me smile. And I feel like it look very similar to it as well. Microalgae are a very diverse group, and we all know, we think of them as microscopic plants. They're eukaryotic cells. Cyanobacteria are sometimes lumped to the same category, but they're sort of different. They're prokaryotic. You can find microalgae in all sorts of environments. It's quite amazing. Freshwater, marine, extreme environments in snow, ice, salt plains, on animals from polar bears to sloths, duck feed. Microalgae feed the entire food webs, aquatic food webs, and they produce at least 50% of oxygen, which I feel oftentimes when we talk about oxygen, we think of trees. But when you think of how much of the planet is covered in water, I feel like algae should get more credit for their oxygen production. How many species are there? Well, if you bring a sample from offshore and you try to identify the species, you think, well, probably too many. And it's true, there are more than 100,000 of species out there. And out of that, only 44,000 are scientifically described and documented. So you have these shelves of books with taxonomic keys, morphology, and we keep about 3,000 in the culture collections worldwide. Which brings me, Hannah, to our poll, if you can please make it happen. Brilliant. So the question I have for you is, how many microalgal species do you think are currently used in commercial applications? This is actually a lot of fun.
Couple more seconds to get people's votes in. Make sure you select an option.
Brilliant. So you're gonna share the results. Okay, there it goes. Okay, so a lot of common sense there. Yes. So the answer is less than 50, which I still think it's quite generous, to be honest. There are a lot of strains of the same species that are used, but commercially, we really use just the tip of the iceberg. And if you look at the vast diversity of thousands of species out there, we really, the whole field of phycology, applied phycology, is quite underexplored and undiscovered. I will be talking about products, Cyanobacteria microalgae products and applications. And this slide, I really put it in here to overwhelm you, to show you all the different products that we can harvest from microalgae, from pigments. Omega-3 fatty acids are very popular for vegans that cannot consume any products from fish. We have pharmaceuticals, protein, minerals, biofuel, everything you can think of. If you're a phycologist, you have to mention Smarties and the blue color of Smarties, which is made of phycocyanin from Cyanobacteria spirulina. So I'll be talking about only about pigments and bioremediation in this talk, but I wanted to show you everything that's sort of possible. So what you cultivate microalgae for also depends on the economic feasibility. So this pyramid is very logical. It has been published in 2013, and it shows the range of different products. So on the bottom of the pyramid, we can see sort of the base commodities where you have large volumes, but perhaps lower quality. And on the top of the pyramid, we have the sort of high value target compounds that you can really sell for a lot of money. So I'm showing you the example here on the right. One is my microalgae slurry that I got from treatment of wastewater, municipal wastewater. And we were very lucky to use that on site for biofuel production. But you can imagine if we were to sell this biomass, it wouldn't be, we wouldn't get paid for it very much because it was grown on wastewater, which limits the applications for it. So for that kind of biomass, less than 1 pound per kilogram. On the other hand, on top of it, I'm showing you red pigment astaxanthin, which is really high value. And the costing for it is from Amazon, from all the places where you can buy a kilogram for 500 pounds if it's 5%. And if you imagine if you would concentrate that astaxanthin to 100%, the cost would be just quite tremendous. There are two concepts that I want to mention to you that should be considered when you're cultivating algae for profit. One is that microalgae change their biochemical composition during their growth. And I'm showing you this classic growth curve where we have on x-axis we have time, and on y-axis we have the biomass or the cell number. And as you can see, the algae kind of have a slow start, and then they ramp up and they start dividing very quickly in the exponential phase. But eventually they slow down because they either run out of nutrients or they start self-shading as well. So they enter stationary phase. And after that, there's the charming death phase. So the biochemical composition changes quite rapidly during this process. So if you just focus on the blue segment of the pie chart, which is the protein, you can see that in the exponential phase, nearly half of the biomass is protein because the cells are dividing rapidly and building up biomass. But as they enter stationary phase, the internal metabolism shifts dramatically. And you can see the blue segment is much smaller, while the pinkish one, which is lipid, for example, increases. I often compare it to us people. When we are young, we have a lot of muscles, we have a lot of protein. As we get older, we may lose the muscle biomass and we may gain more fat. The second concept is secondary metabolites, which are not directly involved in, okay, I just figured out I can move the box with my picture. The secondary metabolites are not involved in growth, and their synthesis is triggered by stress. So when you think about it, microalgae produce these compounds to neutralize the stress. So secondary metabolites are often have the pharmaceutical effects that we want. PhD student Alison Hughes uses this analogy, and I keep stealing it from her, which is about adrenaline in us humans. We don't produce adrenaline all the time, thanks goodness, but we can produce it under stress. So that's what Alison does. She's a student at Strathclyde and with Samson Zantella, and she is mapping secondary metabolites in microalgae. As promised, I'll be talking about pigments and bioremediation. So starting with pigments, I am interested in one group of pigments, which is called carotenoids. They are 40 carbon structures soluble in fat. They absorb light in very specific wavelength between 400 and 550 nanometers, which means they're between the chlorophylls. So they're not green per se. They're more sort of in the yellow, reddish brown, maybe a little bit of green sort of hue to them. And in microalgae, they have two functions. I kind of compare it to Batman and Robin, when chlorophyll is the Batman and the carotenoids are the Robin. If there is too much light that can be harmful, carotenoids harvest that energy and dissipate it as heat to protect chlorophyll. And on the other hand, if there is not enough light, they do the complete opposite. They try to grab any available energy there is and pass it to chlorophyll to continue the photosynthesis that keeps the cell alive. We cannot synthesize carotenoids, obviously, so we have to obtain them from our diet. We have recently read a review about carotenoids, which I feel is like our love letter to pigments. So if you have more questions about that, you can go review the paper. Why do we care about carotenoids? What is so great about them? What are the applications and markets? So because of these antioxidant properties, the neutralizing of free radicals, we use them as antioxidants. We also use them for color and to preserve and stabilize compounds. So the main industries that obviously see the effect of this are food, feed, nutraceutical, and cosmetics. So I'm just gonna run through them just that you appreciate carotenoids a little more. In human food, and I challenge you, if you eat anything orange, look if it had beta-carotene, which is a carotenoid, added to it. I had a peach yogurt in it, and of course it had it in there. Similarly with animal feed, we like to have pink salmon. We don't want to have gray salmon. So it is supplemented to feed to enhance the color. And even with poultry, it enhances the color of the yolk of the egg. So it's sort of nice dark orange, just the way we like it. Human health, this is probably the most important one. Because of the antioxidant properties, carotenoids help to reduce risk of many, many severe diseases. We're all encouraged to eat more antioxidants for our health. And also in cosmetics, there is the protection from UV, there are anti-aging properties, and again, the color for lotions and tanning. So I just want to share this study, which is kind of fun. In Australia, they gave men beta-carotene for 2 months, and they were monitoring their health. And even though they didn't see that much improvement in health of these men, they did see a change in color. So if you stare at this long enough, you may see that the man on the right is slightly more sort of orange, or is more sort of warmer hue. And they took it one more step further, and they let women evaluate which one of these men is more attractive, and women did pick the orange man to be more attractive. So carotenoids can improve your life in many ways, and one of them is the perceived attractiveness. But the bottom line is carotenoid markets, it's a huge industry. It is more than 1 billion in 2016. So there is a lot of money in these markets. One thing I do want to highlight to you that there is a big difference between natural and synthetic carotenoids. We can produce synthetic carotenoids in factories, in the lab, in the chemical process. And of course, natural carotenoids, where you extract it from something living, that is much more difficult to produce. You have to ensure you're taking care of a living organism. So at this point, most of the carotenoids on the market are synthetic. The cost of microalgae-derived carotenoid can be, as we talked about, more than 10 grand, while the synthetic is a third of that. But there is a big but, and that is that the natural astaxanthin is much more potent. It is stronger. There is this publication where they quantify that, and they said it's easily 50 times stronger in its sort of antioxidant properties. And they even advise that maybe we should reevaluate using the synthetic carotenoids if we're not fully aware of how it works and how it functions. So this is our carotenoid project that I wanted to mention to you. There is a specific carotenoid called fucoxanthin. It is this red-brown, beautiful pigment that has all the antioxidant properties as other carotenoids, but it also has anti-obesity effect. And as you can imagine, that is very popular in Asia and Japan. It is consumed as a supplement, as a part of sort of healthy, balanced diet. So I picked two diatoms for our project, Odontella and Phaeodactylum. You can see the picture, especially the Odontella, how packed it is with this brown pigment. And I picked these because very conveniently, they're also on the EU novel lists, novel foods list, which is quite convenient. And what we propose to do is to optimize the production by using specific light. So instead of doing the traditional system where you grow algae very quickly and then you stress them, which the stressing part, of course, it makes them slow down because you're stressing them. So there is always a little imbalance between the productivity and then harvesting the compound of interest. So what I'm trying to do with this project is to avoid this two-step process of growing something and stressing it. I want to grow it in the conditions that it produces more fucoxanthin right from the beginning. I'll let you know how stress-free it actually was. We partnered with Xantella, which is a company right across the street from SAMS that is producing photobioreactors. And we got the funding from Algae UK and iBioIC, which we're very grateful for. The second part of my talk will be about bioremediation. And as you can imagine, microalgae are very effective at removing the typical traditional contaminants like nitrogen and phosphorus. And that's because that's what they consume. That's what they eat. So if we're growing them in the media, that's what we put in. But there are few other surprise compounds, like some of the heavy metals, and even emerging contaminants like pharmaceutical compounds that the algae absorb as well. And that's really fun. They're like little sponges that just soak it all in. So we can use it for the algae to remove these contaminants from the water. By absorbing it, they're removing it from the surrounding water and hence cleaning it. There are three mechanisms for wastewater treatment. One is the adsorption, when the pharmaceutical compound, in my case, the purplish thingy, gets stuck literally on the outside of the cell, so it adsorbs from the cell. And there are several advantages to this process, because you can have dead or alive cells. They don't have to be alive for this to happen. And you can use a variety of passive sorbents, algae, wood chippings, biochar. So it is easy to retrofit to existing wastewater treatment plants, because you're basically just adding a biofilter, something holding a biomass that will passively absorb these contaminants. The second mechanism is absorption. So you are now getting the contaminants inside the cell. And for that, naturally, the cell has to be alive because it requires active transport inside. Theoretically, this could be more effective because you are getting the contaminants not only on the outside of the cells, but also inside. So you could say it should be more contaminants removed from the water. But because you have to keep the algae alive, there may be economic cost to doing that. And the third mechanism is biodegradation, where you break down the pharmaceutical compounds. So it's production of enzyme. And very often, this is a collaboration between microalgae, bacteria, fungi. Oftentimes you see in bioremediation that it is the polyculture and consortia that are used to degrade and break down products. So we have a project that is brewing in the background, looking at removal of pharmaceutical compounds. But the project I do want to tell you about is using byproducts from food and drink industry to supplement microalgae. And what we did is we took actually several powders, but I'll show you just two here. This is a grain from when a brewer is done, there is some leftover biomass, so it's made into a powder, so that's brewer's spent grain, and also tomato powder. And we added this powder into a flask, and we, with seawater, we autoclaved it, removed the solids, and then we ended up with something that I call final liquor. You can think of it as tea. It has color, it has all the leached out nutrients, but it doesn't have any solids. And then we just added our microalgae in it, several species at different concentrations, and we monitored the growth. Do the microalgae like it? Can they benefit from the addition of this powder or liquor? So what did we learn? Well, on the bad side, the liquor can be quite low pH, depending what is the source. But that's probably not preferred, and you can compensate for it. It can also be high in ammonia, which can be toxic at very high concentration. It is so dark, the color of the liquor, that it steals some of the light from the microalgae. And in some instances, it does a lot of foaming as well. And the solubility of the powder can be quite low. On the plus side, though, it is very rich in nutrients. And if we make artificial medium, it has 10 times more nutrients than we would normally put in there. So it is just loaded. It also has vitamins. It has dissolved organic carbon. And my favorite part, that it is food grade. So that opens up the potential application for any biomass we gain from this process. We can apply it to pharmaceuticals or cosmetics, which is something we could never do with algae grown on municipal wastewater. There's also value in the powder alone. There's a lot of energy in the powder as well. So I'm very excited to reuse it in more practical, sort of sustainable way. In summary, the liquor can stimulate microalgal growth, which was quite exciting to see. Not for all species at all concentrations, so it has to be sort of managed with know-how, but it is definitely a nutrient source. And the algae we managed to grow were very rich in protein, so the gained biomass can be used for food or feed very easily. I have to mention other SAMS projects, and we really do a lot of microalgae work. So the other projects would be from Dr. Matt Davey. He's looking at using microalgae for pigments for inks. We have two PhD students, Ellie and Joe. Ellie is looking at biorefinery and high-value products for microalgae, and then Joe is looking at sort of sustainable production of algae for aquaculture. There's also a large project that is finishing on Friday, so we're all just sprinting towards the finish line. Michelle and Mikey are leading this project. The acronym Abacus, I feel like they cheated a little bit from the title. They just picked random letters there. But the project is absolutely fantastic. It started with screening more than 100 different strains and picking high-value compounds and squeezing as much of the high-value compounds from the algae and building up on it all the way to commercial and market applications, which is fantastic to see it from the beginning to the end. And I wanna leave you with where we are sort of in terms of commercial companies in Scotland, because people are often surprised or they don't know everything that we have available here. So we have CCAP, right, our seed bank. We have Xanthella that is making, creating photobioreactors. We have Scott Bio that are successfully producing phycocyanin, which is this blue water-soluble pigment that you can apply to food and feed. We have Microfractionate, Monica and Brian, who are looking at harvesting technologies. So you can start with growing the algae, harvesting them. We have MyAlgae as well, which are using some whiskey waste to grow microalgae for omega-3s. And we also have consulting green scale environmental technology that are looking at wastewater bioremediation. So the future in Scotland is bright, and I will seamlessly pass this to Adrian, who will tell you about seaweed.
I hope it will be seamless. There we go. Is that working?
Yep, you're good to go.
Fantastic. Hi, thank you very much, everyone, for joining today. My name's Adrian MacLeod, as mentioned previously, and I've been working at SAMS for over 10 years now. I started in marine biology, looking at biofouling, renewable energy, and now I've spent the last 5 years working on seaweed aquaculture, and I manage the 2 active seaweed sites that we have at SAMS. So today I wanted to give you a brief introduction to seaweed cultivation and a flavour of what we're working on at SAMS to support the developing industry. And I thought I would do something a little different and try and highlight just 3 key areas of work where I believe we could benefit from wider collaboration within the MASTS community. Okay, so in essence, what we're trying to do at SAMS is to develop the technology and develop the practices necessary for the production of this very promising biomass. And whilst we're looking at the environmental implications and the social interactions, we need to be looking at large-scale aquaculture in order to develop some of the applications that we're working on at SAMS. So why seaweed? Well, it's a very fast-growing biomass. It requires no fresh water or pesticides or nutrient inputs and poses very little conflict with terrestrial agriculture. And this is important because the majority of our food energy actually comes from land-based agriculture. And seaweed's increasingly seen as a source for human consumption as well as for feed and also health products. But we're also looking at the production of chemicals in order to, or the raw materials for many industrial applications. If done intelligently, seaweed can provide ecosystem services through filtering the seawater, and the example of that would be integrated aquaculture. But we can also work with multi-use platforms or design seaweed cultivation systems which work with other industries such as static fisheries. And it's important to say that, globally, seaweed aquaculture is one of the fastest growing aquaculture sectors. And it's been proven in Asia for many years now, but it's not yet being commercialized here in the west of Scotland or the West in general, sorry. And, I'm going to touch on some of the reasons why this is the case. So there are a large number of seaweed species to choose from, and they all have very divergent life strategies and compositions. And I think it's fair to say that we're casting the net quite wide in terms of the number of species that we're looking at. We work on the reds and the greens, and we're trying to get these to the point that we can commercially supply them through our hatchery. But in order to focus this presentation, what I'm going to do is just focus on the brown kelps, as they've got relatively simplistic life cycles, and we're much further ahead in terms of commercializing these species. So the main species we're looking at our site, and the main species that are grown in Scotland, is the wing kelp, Alaria esculenta, sugar kelp, Saccharina, and some of the other kelps such as Laminaria digitata. And as a source of biomass, it's important to bear in mind that the majority of the biomass, the wet biomass landed, is 80 to 85% water. And the dry matter content of the kelps is about 84%. So it's has a rich carbohydrate composition. But there's less protein, roughly around 1 to 10%, and even smaller amounts of lipids. The main structural carbohydrates are alginate, which forms roughly 30% of the dry weight, and cellulose or glucose, which is roughly 10% of the dry weight. And the alginate and cellulose remain relatively constant throughout the year, and the storage carbohydrates, mannitol and laminarin, vary. And so these peak towards late May, early June, and this is when we undertake harvest. Kelps are particularly good at binding heavy metals such as cadmium and arsenic. And it's important to select good sites for growing the kelps. It's also important to bear in mind that whilst iodine is quite high in kelps, which is quite good from the perspective of addressing iodine deficiency, they need to be handled with care to not exceed current safe limits when looking at human consumption. Kelps are rich in micronutrients such as vitamins B12, pigments such as fucoxanthin, and a rich array of seaweed-derived bioactives. And there's increasing medical research looking at bioactives. And so it's an incredibly interesting biomass to work with, but there's a lot of work that needs to be done with regard to developing the products and the processes necessary for developing a thriving industry. So one of the current challenges to developing the industry is the cost of production. And really at the moment, until we get more efficient cultivation methods and develop the cultivation practices, the cost sits at around £2, £2.50 per wet kilo. And what this means is that seaweed aquaculture is largely restricted to high value, low volume applications such as nutraceuticals, cosmetics, and specialty products such as the health food products that you see in some of the health food shops. To get to large scale production necessary for lower value feed, food, and fuel and energy, which are commercially out of reach of using the current practices and the current systems. We need to develop the biology, but also develop the engineering and develop the tools that we use to get to scale. And I'm going to touch on that in a wee bit. One of the other ways we're trying to get around this is to develop the processing methods that can extract the highest value from the seaweed biomass. An example of this is the biorefinery approach, where we can extract the highest value products first and then use the would-be waste streams for energy or lower value commodity products. And in fact, a lot of the biomass we produce at the farm has gone into several European research projects focusing on developing large-scale biorefinery approaches. So let's talk about the cultivation process. So SAMS operates a commercial hatchery, and through our research, we're able to develop the methods needed to seed our cultivation sites and other cultivation sites in and around the UK. And generally, the gametophytes are isolated from the local population, kept in sterile conditions within the lab, and then are grown vegetatively until they're needed. And we have one of two seeding methods. The first one is where the gametophytes are allowed to become reproductive, and then they're seeded onto 1 millimeter string, which is then kept in the hatchery for up to 4 to 6 weeks. And then these reels are taken out to the farm and then wrapped around larger structures. And this is the standard method, and this is the method that is used in Asia, and it produces reliable results. We're also looking at a low-cost seeding method, and this is where we take the same gametophytes that have become reproductive, and we apply these directly onto specialized ropes using hydrocolloid binders. And this process can be automated and taken to scale, and the requirements in terms of the hatchery and the aquarium space and the resources means that we can supply this method at a cost that's an order of magnitude less. So the key areas I want to focus on today is developing this low-cost seeding technique sufficient for high energy areas, and the control of growing conditions. This is about site selection and what we can do to maximize the yield and also the quality of the biomass. And also I want to focus on infrastructure design, because I believe this is an engineering challenge, it's a physics challenge, it's going to take multiple disciplines to work in seaweed aquaculture to address some of the barriers that we're currently facing. So all of these areas I want to focus on are linked because what I'm about to say relates to the surface 2 meters of water in very, very high energy environments. And these high energy sites that are suitable for seaweed aquaculture are highly seasonal, and they're very practically demanding environments to work in. Of course, this is a common problem for any aquaculture activity, but the very processes that make our coastline so productive are the processes that make them so challenging to work in. And so, you know, as we move from hunter-gatherers of the sea to farmers of the sea, we're going to forever come up against this practical challenge. And in Scotland, we tend to overcome this problem by using the complexity of our coastline. But it's going to take a lot of thought and a lot of work to develop offshore aquaculture, for example. Okay, so the seeding technique that we developed along with partners within Europe was originally developed in the European FP7 project called At Sea. And we subsequently moved on to another project called Macrofuels, where we took this process to scale. And I seeded large net structures at the farm, and we ran into a couple of problems. And what we noticed is that when the weeks after deployment were quite settled, we got good results, and the weeks after deployment, which were quite rough, we got poor seeding results. And we tended to find out this information in January, when we would hope to see some growth on the nets. And so what we did was we took this problem back into the lab and used a high-speed flume to monitor how the binder was working in high and low flow speeds, and look at what we could do to develop the binder technology. And you can see from the graphs here that the high speeds that would be typical of a storm event, for example, we saw a huge loss within the first 24 hours. And even at lower flow speeds, which would be typical of any day at the farm, the binder was only working for up to 24 hours. Sorry. So what we wanted to do through a bindweed project, and this project's still ongoing, is to firstly redevelop the materials that we're using for direct seeding. And so we've moved to a rope-based system that will be compatible with a number of different cultivation approaches. And we've redesigned the process, so we now use a multi-stage seeding process. And we're just in this month looking at redeveloping the binder. And the idea here is that we want to develop a binder which provides the additional 7 to 14 days protection that the seed needs in order to get established. So moving on to the next area, controlling growing conditions. The first thing I want to say here is that it's not possible to control growing conditions in the same way that we control growing conditions in terrestrial agriculture. We can only use locally sourced reproductive material, and so site selection, timing, and the selection of cultivation depths are really the tools at our disposal in terms of producing the best results. So with this in mind, what we've been doing at our farm is to try and understand how we can change the timing or the cultivation depth in order to produce the best results. And I've taken this example here of an experiment where we looked at the effect of outplanting time on the seaweed biomass and the yield. And we've done similar experiments looking at other species and looking at the composition, as well as things like the heavy metal content. But this only goes so far, because it tells us how to manage our site, but really to move to the sort of deeper understanding that will support the industry as it moves forward. We need to monitor at our site and also monitor throughout the year through different years, as well as monitor at multiple locations. And so we're trying to understand the effect of the environment on the biomass yield and also the quality. And I've got an example here in the top right hand of the slide, where the selection of the harvest point is really, really important. So this is some data of mannitol and laminarin concentration just approaching harvest. And at this point, the biomass is doubling every 25 to 30 days. And we're also dealing with biofouling that's going to, reduce the quality of the crop very quickly if it's left in the water for too long. So we're deploying monitoring systems at the farm through a project called Impact, and we want to monitor the environmental cues that are needed in order to manage these systems better, so we can select the correct harvest point as well as the correct seeding point and any other mitigation measures that need to happen in between. We're also developing a system at the farm this year, excuse me, that's compatible with oysters, and another project that starts in September called Astral. We will increase the number of shellfish species that are kept at our site. Okay, so moving on to infrastructure design, and this is something that I get super interested in. Being a biologist, I keep coming up against these engineering and sort of physical challenges of working in these environments. This is a beautiful day, and you can see the seaweed being harvested there. But we manage 2 seaweed farms, both are 20 minutes from sands by using a small boat. And our largest farm, which you see here, has the capacity for 2.4 kilometers of seeded material. It's quite a low stocking density because we're undertaking experiments, both medium scale and large scale. But we also work with local businesses, and this is relatively small site compared to what we would need to get to for a commercial scale site. So it's a 100 by 100 meter hectare site. Currently, there's a huge range of different cultivation systems, from long lines to grid-based structures like these that are being trialed throughout Europe, and each has their pros and cons, and really the physics of the site and the engineering and the biology need to all be tied together to produce low cost, easy to maintain sites that are also productive. And so some of the things we're looking at SAMS is to develop or optimize the stocking density of the site so we can utilize the primary resource, which is the sun, in the most efficient manner, and also working to reduce the amount of plastics and other materials that need to go into the cultivation process to try and reduce the environmental footprint of that system. Future cultivation systems might also need to think about how we design a cultivation site so that it is compatible with other marine users. And we're working with a local creel fisherman to understand how we might be able to redesign the system so that he can fish within the same space. So there are a number of really interesting things to look at in terms of the cultivation design, and we will need an efficient process if we're going to meet the low-cost commodity-based products. So I'm just going to summarize here. There's increasing demand for seaweed, both within Europe and globally. And this creates a number of interesting opportunities to develop this resource and develop jobs within the area. But we also need to think about the social license constraints and work with local stakeholders to design systems and methods in a way that's compatible with other activities. I spoke a little bit about controlling the growing cycle, but this is incredibly important because we need to control the quality of the product for many of these high-end applications, for the high-value foodstuffs, for example. And that's really where the expense of seaweed aquaculture is justified if we're able to attain that high quality. And I spoke a little bit about the automation required and the need for easy and automated seeding and harvesting processes. But there are some barriers in the way, and we are doing work to try and address some of these. Site selection is really important, and understanding the carrying capacity of the receiving environment. So that might take the development of models and other tools which will allow us to easily answer the question of where we put these structures and what scale we can get to. Conflict with other marine users is really important. I've mentioned that previously, and also the management of foreseeable problems. I mean, we can learn from existing aquaculture and do something about or remain vigilant about the foreseeable problems such as disease and pests, and take the necessary action that's needed. Design, we spoke a little bit about the need to develop low-cost, efficient systems, and that's going to take lots of input from lots of disciplines and hopefully the wider MASTS community. So I think that's all I've got time for now, but just to say, please get in contact with me if you have any questions or you want to discuss any of this, or any of the wider team. We seem to be growing daily, the number of people who are involved in seaweed aquaculture. And I'd love to thank the funders, without which this work wouldn't be possible. So I think I'll stop there and include a picture of the site under normal conditions.
Thank you, Adrian, and thank you, Lucie, for being today's MASTS webinars. For all our attendees, please submit your questions using the Q&A box that is at the bottom of your screen, and we can work through those questions that you have for either of our presenters, or something that maybe both of them can weigh in on if you want to ask a question. Alternatively, if you're watching on YouTube, please just submit a question that is at the bottom of the screen, and we'll be able to answer that question too once we get to it. So, Adrian, you've spoke about, there's lots of purposes for seaweed, and one of them that I know about, don't know anything about seaweed or algae at all, is that, it's sometimes included in food. And I was wondering, there must be some food safety guidelines for this. You can't just grow it anywhere, am I right? In that, there's a whole variety of things that could be in our waters. So I was just wondering if you could just expand, how do we know that the seaweed that we have is okay, and that is not, where it's growing?
So the Food Standards Agency is sort of developing the guidance on including food as a foodstuff. The European Commission also has, guidance on the amount of, for example, iodine or heavy metals that can be included in a daily portion of whatever product you're producing. But the industry is still catching up a little bit, and through the Scottish Seaweed Industry Association, these are discussions that are ongoing regarding food safety. And so I think whilst there's some guidance out there, there's more guidance to come in terms of how you look after yourself when you're eating seaweed. But it's fairly safe.
Yeah, so we've got a couple of questions that have already just pinged through. So the first question is from Fiona, which is for Adrian, asking about that seeding capacity you mentioned at SAMS. Obviously this is a lot of projects that's still happening and you're still understanding what is best for your seaweed, but at the moment, what's the seeding capacity that you're experiencing?
In terms of the volume that we can get to within the hatchery, I don't have the figures in front of me on the volume we can get to. It's something along the lines of maybe 10 kilometers is what we can seed in one run. And so that would be a 6-week period where we'd be growing up 10 kilometers worth of seeded twine. And again, this kind of highlights the need to develop the direct seeding method so that we could get an order for 100 hectares of seed, and we could just turn that around within a week or two. So hopefully that answers the question that was put forward.
Yeah, Fiona, if you have any other questions, please pop them in. Next question is from Gracie, again for Adrian, asking just to remind us, what were the red and green species that you are working on, and are you interested in working on any intertidal species?
So I'll answer the first part of the question first. So we're really interested in Palmaria palmata. It's got a fantastic flavour. It would be a high value product. It's got a complex life cycle, and we've done a lot of work to look at how do we get the seeding fragments in the lab at scale that we could seed a farm. But we're not quite there yet. We've run into a few issues with regard to parasites that are inside these reproductive stages, and so working at ways to get around that. The other side of things would be the alva, the green species alva. And I say alva without giving you the species name because there's lots of different types of alva. But basically, green alva would be a summer crop that we are super interested in developing. And the more types of species that we can grow at the farm means that we could increase the harvest frequency and also have year-round cultivation. So alva would grow in the summer, and the reds and browns would grow throughout the winter and spring.
Really trying to maximise on the Scottish weather with the seaweed. So talking about actually the weather and sites, our next question from a different Fiona is asking about site selection versus important, and what method is used to identify what method to identify sites would you say is the most effective GIS?
Oh, that's a good question. So I use the information from the Scottish Government website as a starting point, but actually it highlights another important area. Seaweed cultivation at the moment requires the same kind of space as you would normally put, say, salmon aquaculture. You can't go too shallow, otherwise your farm will end up on the shore during a storm, and you can't go too deep because it'll be too expensive to moor that structure in place. So in terms of site selection for the browns, we need places that obviously are full salinity. So we don't want places that are influenced by fresh water input during the winter months, as that would kill off your crop. We need places that are got a lot of light, and so sedimentation or sediment in the water, needs to be low, and the mooring and practical considerations there are also important.
Okay, thank you. Next question is from Owen, asking about your production costs. You say that they're around £2 to £2.5 per kilo. Could you potentially try and break that down a bit more, please?
Okay, so the first thing to say about the production cost is that is something that we are doing more work on through a project called through work with our through SRSL, but through discussions with other people, £2, £2.50 per wet kilo is roughly what people are talking about. And I believe that is, if we were doing this in anger, just going out for commercial purposes, that's how much it would cost us at the moment using the current methods. But I'm not going to give you a breakdown of that. I would say if you're using twine seeding, roughly 30%. A third is seeding, a third would be the cultivation equipment, and a third would be harvesting and processing.
I see, thank you very much. A question that I have for Lucie, if Lucie could just unmute herself. So as I've already said, I don't know anything about algae, and you spoke about those 3 ways to do bioremediation for such as pharmaceutical compounds, and you gave those 3 examples of how that can be done. And I want to know, is that can that be done, those three methods, to all algae out there, or does some just work better with others, or is it just, yeah, is this those three methods can be applied to any algae that we're interested in using?
Yeah, thank you, Hannah, for asking a question about microalgae. Yes, so the methods, we usually sort of don't care as much about how it's done, as long as it gets done and the pharmaceutical compounds get removed. But you're absolutely right that there are some microalgae that can be more sticky, and it depends on the covering of the cell, because there's such a diversity. So sometimes they have, coccolithophores have shells, sometimes they're more, carbohydrates, so that makes them more sticky. So yes, different species have different efficiencies in removing contaminants, yes.
Great, thank you. Adrian, there is a question from Fiona asking, what are your thoughts on offshore co-mining, such as wind turbines?
Yeah, I think it's a great idea. And again, it comes down to sort of the kind of engineering of how would you go about that. I would say that putting seaweed aquaculture offshore could be quite demanding. And one of the things that people would need to look at is how many days at sea are you going to be able to work the site during seeding and harvesting? And I think that will be a critical factor in the decision there, because the larger the boats that you use, the more expensive overall the cost of production is. And really we want to use smaller boats, so therefore we can't go into areas that, half the time in the end of May, you can't access the site and actually do the work of harvesting. So some big challenges there, but certainly if they can, combining these two activities reduces the overall cost, then fantastic. Absolutely.
Excellent. So the next question kind of relates to this, asking, have you done any work with wind farm companies to explore this?
We have, from a social license perspective, but I wasn't personally involved in that work. But many of our collaborators who are situated around the North Sea are working with wind farm companies that, exploring this potential application. But I would just like to maybe mention that, in Scotland we have this real advantage that we've got this complex coastline. So there is ways to cultivate seaweed without even having to moor the structure from one landmass to another, for example. And so there are other ways that we can get around the cost of mooring the structure, which would also be along the same lines of offshore wind.
Yes. So from, my goodness, we're getting so many Fionas asking, a different Fiona asking this question. How does market size compare microalgae versus macro, and where do you both believe the greatest growth to be?
Lucie, I'm going to let you answer that.
I was waiting who's going to send us against each other. Yeah, no, I think, and also the next question, sort of, which one is more profitable, or would you combine them? I think they both have very different sort of advantages. Obviously, seaweed, the harvesting, there is nothing, you cannot harvest microalgae as easily, so that's a good advantage. In terms of the size of the market, I think that is difficult for me to say. For microalgae, there are some products like the astaxanthin and Haematococcus production that are just worldwide, and there's really a lot of money in it, but those are only selected products. So I think if all the potential would be harvested, I think it's kind of difficult for me to compare. I don't know, Adrian, if you have better response to it.
Well, I would just, from a seaweed perspective, the market is globally large. There's up to 30 million tons of just Laminaria japonica that's grown annually. So the market's there globally, but in Scotland there's not a huge market pool at the moment. And this is because companies need to solve so many problems. They need to solve the seeding, the harvesting, the processing, and as well as developing the market, developing the product. There's a few. So yeah, it's going to take a lot of people working together to develop that market in Scotland.
Thank you both for that answer. So regarding the research that has happened on seaweed aquaculture, is there any available that is open source? So for instance, one of the questions that has come through is asking, are we able to access this research so that we could potentially apply this to a commercial seaweed business plan?
Yes, you'll notice in my slides that there was a few comments underneath some of the data saying unpublished. It's our plan to catch up and publish these results. So we would aim for open source publication as well. So that would be what I would say to that question.
Yes, I think that's a really great point to end on, because if anyone has any other further questions, they are more than welcome to get in touch with either of today's presenters. The emails are on your screens right now. Do we have any other questions coming in? Oh, someone's just asking about that open source information about research, asking would they be able to share it through the SSIA membership? I'm not sure what that acronym stands for, but I'm sure one of them must stand for seaweed.
Yes, the Scottish Seaweed Industry Association. Yeah, can do.
Excellent. So thank you both so much for today's wait, have we got another? Yeah. Thank you both so much for today's webinar, presenting alongside each other, providing the whole spectrum of algae that is the potential of it in Scotland. So thank you very much, Adrian, and thank you, Lucie.
Thank you.
Thank you, Hannah, for inviting us.
To all our attendees who are still with us, I would just like to point out the upcoming talks that we have left in our MASTS webinar series. We are ending on the 2nd of September because the MASTS annual science meeting is on the 5th to the 9th of October. Abstracts call is now open. So if you want to talk at the MASTS annual science meeting and present to a whole variety of attendees from around the world, please make sure that you check out the MASTS website about that. But if you want to attend any of these upcoming talks, please make sure that you register using the same registration link that you have used to attend this very talk. We look forward to seeing you at any of our other webinars, and please join us for the MASTS annual science meeting as well in October this year. Oh, I should say it's online.